Hello, I am Mang from Wageningen Food Safety Research, and today I will update you briefly on the progress of Task 2.4, the development and validation of lateral flow devices, in short, LFDs. I will first talk about some adjustments and optimizations of our homemade LFDs, including switching the reporter to carbon nanoparticles, excluding tritone from our running buffer, some of our own neonicotinoid single tests developed independent from our Chinese partner. Uh, Also, some new ideas in quantifying the LFD readings. Then I will share our total neonicotinoid profiling work with you using our XMAP technology. And since Anne is not able to join us for this consortium meeting, she had kindly asked me to inform you about our collaborative attempts in the development of LFDs for the detection of B viruses. Finally, I will show you our plans for the next 6 months. Previously, we have presented these WFSR homemade LFDs for the detection of 4 bee-harming pesticides, namely chlorpyrifos, ivermectins, fipronil, and pyrethroids. We have reached the sensitivities ranging from 0.1 to 10 ppb, which is quite ideal. In the running buffer of these LFDs, a surfactant Tritone X100 was used. However, at the beginning of this year, we were informed that Tritone X100 is no longer allowed in the EU due to environmental issues. So we were left with 2 options. The first is to kick out Tritone completely from our buffers. The second is to try to find a replacement for this surfactant. I will show you what we did with the example of chlorpyrifos LFD. Our running buffer before, which was our standard running buffer, was a 100 millimolar borate buffer, pH 8.8, with 1% BSA, 0.05% twin, and 0.01% tritone. Before we had a sensitivity at 1 ppb. To remove tritone completely, we also have another buffer in our lab, which we use for some other LFDs. It has 10 times more twin than our standard buffer. This percentage is very rare in literature, but it works in our hands. And as you can see, at 0.1 ppb, the line also disappeared, so that brings our sensitivity 10 times higher. However, if you look at the edges of the line, they look a bit diffused, so it's not that visually appealing to customers. After doing some literature research and consultation with customer service from Sigma, we were recommended with this Tritone. It is also a surfactant which has a similar molecular weight and function. So we replace it 1-to-1 in our standard buffer. After testing, we can see that if you compare with our old standard buffer, the sensitivity does not change that much, except at 1 ppb you do see a slight test line. So we think that is maybe just a little bit less sensitive, but it's not that significant. You can debate about the appearance of these strips. Depends if you're aiming for a clearer background, less pink background, you want to go with the high twin buffer. But if you are aiming for more or less the same intensity between the control line and the test lines, then you can go for the Tritone buffer. You might have noticed that the sensitivity of our chlorpyrifos LFD had gone down from 0.01 ppb to 1 ppb, and that is because we adjusted the determination method of the concentration. Before, we were talking about limit of detection, and the concentration that showed was in the entire assay, that is 1-to-1 ratio. However, when we were trying to introduce matrix into the assay, which means that we have to dilute and we want our limit of detection to refer to the concentration in the sample or extract. Usually we use 1 microliter or 10 microliters in 100 microliters assay, and we're only referring to this proportion. Accordingly, we adjusted the sensitivity that which we presented before to the 1 to 100 or 1 to 10 ratio, and we have the sensitivity listed in the table. The next update that we have for our LFDs is we switched our reporter molecules from polystyrene beads to carbon nanoparticles, mainly because carbon nanoparticles are more consistent between batches, which saves us a lot of time and repetitions for one experiment. Carbons are also smaller in size, so when we run the assay, the lines are always shown sharper. And in some assays, they're even more sensitive now with the carbon nanoparticles. The disadvantages of carbon nanoparticles is mainly the color problem. Before, when we used polystyrene beads, we can make the assay look red, blue, or green, but with carbon, it's only black. And we also noticed that when we prepare LFDs for dedicated for carbon reporters, we noticed that we have to use 4 times more sparing material on the test line. Here is the comparison of assays developed with polystyrene beads and assays developed with carbon nanoparticles. I've updated the sensitivity according to the sample dilution. For abamectin, which belongs to the ivermectin group, the sensitivity did not change when we switched to carbon. For fipronil, we can see the increase of the sensitivity from 1 ppm to 1 ppb. This is also thanks to a better batch of antibodies from our supplier. For fenpropathrine, a member of pyrethroid, the sensitivity remained the same. For chlorpyrifos, the sensitivity increased tenfold when we switched to carbon. And also in many of these assays, you can see the lines are much sharper and more defined when you compare with polystyrene beads. I believe you all remember this dual-channel neonicotinoid LFD prototype developed by our Chinese partner, Zhejiang University. We have done some extensive validation work on this neonicotinoid LFD prototype in many different matrices, and we were able to detect 6 out of 8 neonicotinoids using this device. And this year, we also requested the materials used for making this device, namely the neonicotinoid antibodies and the neonicotinoid conjugates, to develop our own LFDs. We're aiming to sensitively detect the traces of the 5 neonicotinoids which were once allowed in the EU, especially the 4 neonicotinoids which are now banned. So this is what we have achieved. We developed single tests for acetamiprid, imidacloprid, thiamethosam, and thiacloprid with limits of detection from 1 to 10 ppb with the sample dilution of 1 to 100. We can even increase the sensitivity by 100 times if we're detecting neonicotinoids in water, which we can then dry the materials on the strip and then directly measure the water, so that will give us even higher sensitivity. We're also now working on the clothianidin LFD, which we don't have yet. And compare with this dual-channel LFD prototype developed by our Chinese partner, they use colloidal gold as their reporter, and we use carbon nanoparticles. And the reporter that we use is definitely much cheaper. So if we can manage to produce such prototype, we are happy to provide them to be good partners and all the apiaries for free. So other than the visual reading and thereby qualitative interpreting your LFD results. We're also working on how we can quantify or semi-quantify the LFD readings. In the lab, we have a flatbed scanner that can scan the results in a relatively high resolution, and we can determine the intensity of the bands using this total lab image analysis software. With some calculations, we can calculate back from the intensity to the concentration of our analyte. Before when we were presenting the fipronil results, we have shown you this cube reader. It is a handheld device you can take on site easily. You can develop your LFDs on site and then insert your strip into this box, and on the small window you will have the concentration of your target. It seems like a very handy device, but it didn't get too popular in our lab because it involves heavy programming. So we think it is more suitable when we have more standardized tests that we can use very frequently. Then it will be worthwhile. And this year, a PhD student in our lab used the 3D printing technology to develop this holder for your smartphone, where you can also develop your LFDs in. And with the built-in camera of the smartphone, she can take pictures of the LFDs or even record a video of the development and quantify it using also image analysis software called ImageJ. And this paper has been published recently. The smartphone ImageJ analysis of LFDs is actually very powerful. Let's take the fipronil LFD for example. On the left, if you look at the strips with your eyes, you would say that the sensitivity of this test is around 10 ppb. However, if you log in the intensity of the test lines of the strips and normalize it against their control lines, you will have the TC ratio, and you plot it against the concentration, and you can see a clear difference between the blank, which is in red, and the 0.01 ppb dot already, so that increases the sensitivity 1,000 times. Now I will continue to show you how we utilize the neonicotinoid antibody and the conjugate material that we got from China. We have developed a total neonicotinoid profiling assay using our benchmark XMAP technology. This is a small innovative project on the edge of Be Good, and we have used a fraction of this assay to assist the measurement of the field materials during the collaboration with University of Coimbra. This assay is a bead-based suspension array. The beads are shown as the large red dots, and the neonicotinoids are represented by the small blue dots. The Y-shaped molecules are antibodies. It's either the blue ones are specific for neonicotinoids, and the black ones are the species-specific antibodies. The RPE, the fluorescent reporter, is represented by an orange fluorescent dot. And by incubating the neonicotinoid coupled beads, your sample and the antibody together, there will be a competition for the antibody between the beads and also the free neonicotinoids in your sample. After washing, you will have this bead, antibody, and RPE complex that can be measured by the MagPix planar array reader. And this is a competitive format, which means if you have more antibodies present in your sample, there will be less antibodies bind to the beads, and therefore you have a stronger inhibition during the assay. The MagPix planar array reader is a fluorescent imager. It has two LED to emit lights to excite the molecules, and also two CCD cameras to capture the signal. The red LED light can excite the beads, and the signal is captured to identify the beads because the beads are color-coded. The green LED excites the RPE molecule on the secondary antibody, and this will show us the actual signal during the measurement. So with this XMAP technology, we have developed 5 individual assays for profiling of all 8 neonicotinoid targets. And we have validated our profiling assays in 4 bee-related matrices, namely tap water, bees, pollen, and honey. And we're very looking forward to send this manuscript for a scientific journal to publish as soon as possible. Now I would like to move on to our collaborations with Task 2.5 INRA in the attempts of developing lateral flow devices for the detection of acute bee paralysis virus, ABPV. At the beginning, we thought we could work more closely together, but unfortunately, Corona crisis had prevented us from traveling until now. So our solution was that Andamon from INRA can ship us the materials, and then we use that to develop lateral flow devices in WFSR. She has had isolated ABPV virus from infected bees, and she also provided us with the polyclonal antibodies that she raised against the ABPV peptides. And we received these materials in good order, and we first tried a sandwich assay format, which is to capture the positive ABPV between 2 antibodies, and that will give us the signal. Unfortunately, this sandwich assay format always gives us false positive. And then we tried also the direct competitive format, which is more or less like what we do for the pesticides. This is not ideal, but we also tried it, and it keeps giving us false negative results. We have spent a lot of time in the literature research and trying out in the lab, but we still couldn't really solve this problem at the moment. We were thinking that it's probably because of the glycerol that is present in the antibody solution for the preservation purpose. And we're trying to tackle this the next. To tackle this problem, Anne had sent us a new batch of this ABPV polyclonal antibodies, but this time glycerol was not used in the buffer. We hope this will give us some positive results. Alongside ABPV, we're also going to work on the chronic bee paralysis virus, and the antibody for CBPV was raised against the full virus. So I think with this method, we will have a broader recognition of the virus particles. Before we were not able to travel, Anne could not come to our lab to join us for the development of LFDs, but we have, as the borders are now opening up, we have agreed that she would visit our institute in late July. We will benefit a lot from this collaboration, I think, and especially her knowledge in virus would be very helpful for our development of ABPV, CBPV, LFDs. We also have a few other things planned for the coming 6 months. First, we want to standardize our homemade LFDs for the detection of pesticides and applying them to more bee-related matrices. We also want to put at least one LFD into a cassette. It sounds like an easy work, but it's actually a very delicate job. So in the end, we would like to have at least one working test in the cassette, which you can take on site with you, just like the Neonicotinoid dual channel LFD prototypes that is developed by our Chinese partner, Zhejiang University. Speaking of that prototype, we're very happy to assist and help arrange these prototypes for the pesticides monitoring in Tier 1, Tier 2 mini apiaries. Also, we want to implement more this smartphone image J analysis for quantification of our LFDs. In late May, we held two LFD workshops, and you have probably read it from the newsletter. It was a great turnout, and we're looking forward to have more workshops with you, more in person, and also because travel ban within EU is slowly being lifted. So we're also looking at more collaborations. Please feel free to contact us if you think we can assist you in any way.